Let me introduce the next speaker, who is Dr. Peter Loskill. Dr. Peter Loskill is a W3 professor for organ-on-chip research at the Everhard Karls University, Tübingen, and the Natural and Medical Sciences Institutes, as well as the Vice Chair of the European Organ-on-Chip Society. He received his PhD in physics from Saarland University. He currently heads the Microorganolab Lab and the 3R Center Tübingen for in vitro models and alternatives to animal testing. The 3R Center Tübingen aims to provide scientists with low threshold access to novel alternative methods to animal testing. In 2015, he was named as one of the Technology Review's innovators under 35 Germany and awarded a Fraunhofer Attract Starting Grant. His talk today will focus on organ-on-chip models, recapitulating complex human immunocompetent tissues. Okay. Yeah. Let us welcome you.
Thank you very much for the introduction. I hope you can hear me well. First of all, I'm very sorry that I couldn't join you. I would have loved to travel to India to join you and give the talk in person. However, I couldn't do it this time, but next time I'll definitely try to come. I'm happy to present the research we are doing in terms of organ-on-chip models, and I'm going to give you a short overview of what we are doing in my lab. Let me start with the definitions of what I mean by organ-on-chip and microphysiological systems. In terms of microphysiological systems, we typically categorize into the two categories, the organ-on-chip systems and the organoid models. Organ-on-chip systems are more the engineering approach of recapitulating an organ microenvironment and the organ's dynamics, functionality, and pathophysiological response. The organoid is more the, let's say, the developmental biology approach. And both is something we count as microphysiological systems. And I'm going to be talking about the organ-on-chip, but I'm also going to show you how we use organoids in organ-on-chip systems to bring them to the next level. For us, important is for the organ-on-chip systems that it is an interdisciplinary effort. So it's not a microfluidic system which is being developed somewhere and then the biology being done somewhere else. We really try to be at the interface of the microfabrication in microfluidics, and on the other hand, the biomaterials and the stem cells and the organoids. And what we're always aiming for is to have a system which has a human tissue, which is very important for us, an in vivo structure and function, a microphysiological environment, and a vasculature-like perfusion. And over the past decade, we have worked on a wide range of different models, and we have now prototypes of models at different levels of more than 10 different tissues, and here you can see some really ranging from brain and retina all the way to pancreas and cartilage. The idea is really to bring together the biology and the engineering and to then translate these systems into applications, and that's for us mostly pharmaceutical R&D, but also clinical research and mechanistic biomedical studies. And instead of going now one by one through all of these different models, I'm going to take you on the journey we take to develop such a model system. So for us, the journey of an organ-on-a-chip model always starts with the idea. And the basic idea is always that we use the human body as a blueprint on the one hand, as a structural blueprint, and also as a cell source. Then we combine these things into a microfluidic platform, and that's then the organ-on-a-chip system. So that's the basic idea. And the next step is then the implementation. Here we follow two different paths initially, biology and engineering, and we have the design, the fabrication of materials on the one hand, but also the cell sources and the tissue preparation, biomaterials on the other hand. There's never a kind of a distinct path, and it's always an iterative approach. So we circle back through these different aspects. And then we come up with a way of generating a tissue, of injecting a tissue, which then leads to the development of readouts, assays, and then a functional validation. And only if we have done that, we can go into applications. And as I mentioned, I want to take you on this journey now, showing a couple of the examples of our model systems. I'm starting with the idea, and the idea is always has one basically underlying concept, and this concept is compartmentalization. So all of our systems are based on compartmentalization, where we separate the transport processes, the convective transport to the media channels, and the diffusive and active transport. Then we have tissue chambers which are tailored to each organ and which have physiological biomechanical properties, but also give a controlled and reproducible microenvironment. These can be individual tissue compartment, but it can also be multiple tissue compartment separated by different types of structures. So that's the underlying idea. For the fabrication, I'm not going to go in detail here. I'm going to show you more the examples, but here we use different types of materials. We focus a lot on PDMS-free materials using thermoplastic elastomers, for example, but also integrating bio-inspired membranes such as electrospun membranes. I want to take you now to the next step, and that's the cell sources. For the cell sources, we either use primary cells or we use iPS cells, because we always want to have a human cell because we're aiming for a human model. Also, we want to get the same cell types from the same donor to have autologous models. So in terms of primary cells, for example, we work with adipose tissue a lot, and I'll show you examples of that. But here we're getting biopsies, and then from these biopsies, we can get the adipocytes, we can get the endothelial cells, we can get the stromal vascular fraction, which are immune cells and progenitor cells, which we can also separate. And then we can get from the same patient blood and get immune cells as well. Then, on the other hand, we use iPS cells, and this is, for example, what we use for our retinal models. Here we can differentiate then iPS-derived RPE, retinal pigment epithelium, organoids, or endothelial cells. So these are the two cell sources. And if you have now the technologies and the cell sources, the next step is really to generate the tissue. And here we're using two basic approaches. And these two basic approaches are on the one hand the bottom-up approach, where we use single cells to build complex tissues, and the assembly of building block approach. And here we merge organoid and organ-on-chip technologies. And I'm going to show you that a few of these approaches. The first one I'm going to start with is the adipose on chip. As I mentioned, we can isolate all of these different cell types from adipose tissue, because adipose tissue is not just adipocytes. It's really a complex mixture of cell types, including the endothelial cells, the progenitor cells, the tissue-resident immune cells, and the circulating immune cells. So we isolate all of them from a biopsy, and then we bring them into a chip. And this is the adipose chip. And we bring the adipocytes, the endothelial cells, the adipocytes and the progenitor cells and the tissue-resident immune cells into a cylindrical tissue chamber embedded in a hyaluronic acid-based hydrogel. And thereby we can recapitulate really the structure of the adipose tissue, but we can also see that the macrophages are migrating around through the tissue and even building something like crown-like structures. The endothelial cells are then added on top of the channel lining, or on top of the chamber lining the membrane along the channel. And last but not least, the immune cells, circulating immune cells of the T cells are being perfused through the system. This approach, or this bottom-up assembly approach, now allows us to build models that are tailored to a specific question. That means we have a modular mix and match toolbox. So we can have either adipocytes or only endothelial cells in the system where we have a less complex, but let's say higher throughput system, or we go all the way to the system where we have all the different cell types. And this allows us really to dissect mechanisms. And I'll show you later how we do it. So this is an example of the bottom-up approach. Another example of the bottom-up approach is our choroid-on-chip system. And the choroid it's basically the outer blood retinal barrier. It comprises epithelium, endothelium, and stromal cells. And the choroidal immune cells are critical for retinal homeostasis. And this is how the model looks like. It's a microfluidic platform which has 3 different channels. And if we look at it from the side, we have the 3 channels. One is the RPE channel, the endothelial channels, and the stromal channel. And the stromal channel or stromal cell types are mostly melanocytes. So we now add microvascular endothelial cells in the top channel line. We add retinal pigment epithelium cells in the top channel. They line the channel. You have a very nice pigmentation, which you can see in the bright field. You have a nice zoneoculans expression, and you have a melanin production. Then we use the microvascular endothelial cells, line the central channel, and thereby have a blood vessel-like architecture. And last but not least, we add the melanocytes in the lower compartment embedded in a hydrogel. And the advantage here of using this bottom-up approach is that we can vary the melanocyte density. And this was actually a study where contract by a pharmaceutical company, and they wanted an analogous to a cynologic monkey, which has a very, very high melanocyte density, and a human system. So what we could do is we now could play around with the density of melanocytes and thereby build models that recapitulated these two different generations. And last but not least, we could then add the PBMCs, or in this case the T cells, and study in this case the recruitment of these T cells when we tested different types of bispecific antibodies. So these are two different ways of using a bottom-up approach to build a complex tissue. In terms of the building block assembly, I'm going to show you something about our retina-on-chip model. The retina-on-chip model, for the retina-on-chip model, we choose this building block approach because when I saw the structure of the retina for the first time, I was almost sure we could not do that with organ-on-chip technology. It's an extremely complex, stratified, multicellular tissue, and you have lots of different cell types that have to be in specific places and interact specifically with each other to actually make up the function of the retina. So what we decided, we're going to combine retinal organoids, which comprise already most of these retinal cell types, and have already a very physiological layering. However, we are missing two important factors, and one is the perfusion, and the other one is the retinal pigment epithelium. So what we then did, we added these organoids into a platform, which we call the retina-on-a-chip platform, and this platform has 4 microwells. And in these microwells, you can add an organoid, but you can also add a retinal pigment epithelium. And you can then perfuse the system from a choroidal-like vasculature underneath the RPE, which you can again line with endothelial cells. So in total, we have more than 7 different cell types, all derived from the same iPS cell, and now can recapitulate the entire structure. And that's how it works. So we first seed the RPE. We culture the RPE 7 to 14 days till we have a very nice physiological structure. So we have a pigmentation, we have a microvilli formation, we have a polarization, and then we add the organoid on top. And crucial was in that case that if we just place the organoid on top of the RPE, the organoid would disintegrate, it would grow out, the photoreceptors would grow out, and we would actually lose the structure of the organoids. So we would not anymore have this layering that we actually needed. So what we had to do a very thin spacing between the organoid and the RPE, and we managed to do that by adding an ECM-like hydrogel in between. And then what we could see that after a couple of days, the organoids, more specifically the photoreceptors on the outside of the organoid, would grow out segments, and not in only individual segments, but really large segment clusters that would fill the entire spacing in between photoreceptors and the RPE. And the interesting part was that if you have an organoid in a dish, or if you just place this organoid in the chip without having the RPE, you will also see segment growth. But you have a much higher number of outer segment-like structures, or in general segments, if you have it in a proximity culture to RPE. So you really have an induction of this segment outgrowth by the RPE proximity culture. All right, so that's one example of a building block approach. The other example for the building block approach is our pancreas-on-a-chip model. And here we're not using organoids, but we're using pre-aggregated spheroids. And these so-called pseudo-islets can then be captured in a system which is tailored for exactly this size and via hydrodynamic trapping. So this was the first generation of the pancreas on a chip, and we actually could get a nice viability of the system. We could see a good insulin production capability, but we realized this is not really the structure of a pancreas to have individual islets, pseudo-islets sitting somewhere in the chip. So we went further and now have the second generation of the pancreas in a chip system. And it looks like that. So we have a multi-layer system. We're in the top channel, we have a media channel, which we can perfuse. Then we have the bottom layer, we have a tissue chamber, which is a dead-end channel. And then in between, we have a porous membrane, which has traps in which we cut into the membrane. And these traps are in a range or in a size range that is a little bit smaller than our pseudo-islets. And what we can then do is we we just add pseudo islets into the inlet tips, and by hydrodynamic trapping, these islets are then guided into this trapping structure. And you can see in the video how they just flush in and then are placed right on top of these trapping structures. And so this is already allows us to have a much more defined way of adding the islets. It's very simple and not manual, so you can just pipette it in. And then we can also add an extracellular matrix. So we then inject into the same tissue channel, we inject an extracellular matrix. Here we used fluorescent beads to actually visualize it. But instead of fluorescent bead, this extracellular matrix, hydrogel, can also include, for example, different type of stromal cells. So we can add stromal cell types around the islets in this case. And that allows us, again, to have islets with a very good viability, physiological function, so we have a good insulin production capability. And if we, for example, add endothelial cells, you can actually see how these islets are getting vascularized. So these are two examples of our building block assembly approach. So in general, we have two different approaches to build complex multi-cell type tissues. However, there's one aspect which I haven't talked about, and that's the density. Most of the tissues in our human body are consist, looking at the volume, almost entirely of cells. You have the extracellular matrix around it, but the majority of the volume is taken up by the cells. In tissue engineering approaches, it's often the other way around. You have hydrogel and you have some cells in between. It is very difficult to actually achieve very, very high densities or physiological densities in a microfluidic chamber if you use post-mitotic cells that don't proliferate. And so we, especially for our cardiac models where we have post-mitotic cells, we had to come up with a way of generating this multi-cell type tissues with very high physiological cell densities. And we came up actually with two ways. One is a bottom-up approach, and the other is the assembly of building blocks approach. And I'm going to show you that now. So the bottom-up approach uses centrifugal forces. So what we do, we now have our microfluidic chips, and we have the ports, the inlet ports, not on top, but from the side. And what we can then do is we can add a cell suspension into these inlet ports, put the entire chip in a Falcon tube, put the Falcon tube in a normal centrifuge, and then we start the centrifuge. And through the centrifugal forces, the cells are being transported from the pipette tips into the microchannels. And because the microchannels are in an angle to the centrifugal forces, but the tissue chambers are parallel to the centrifugal forces, the cells will first go into the main channels and then sequentially fill the tissue chambers in a 3D manner. And thereby we can achieve very, very high density structures in the shape of the tissue chamber. And we can do that actually for 1 or even 8 different chambers at the same time. The other approach for generating high density tissues is a building block assembly approach. And here we first form monodispersed spheroids to have a very controlled and defined starting point, and also a defined cell composition, so we can mix different cell types in these spheroids. And then we add these spheroids into a chip where we have a small constriction, which then captures these spheroids. And that's how it looks like. So they pop in there. Again, we just need hydrostatic flow, pressure. So there's no kind of a manual pressure buildup or anything. So we just add a pipette tip with these spheroids into the inlet ports, and then they will pop in there. And within 24 hours, they will then start to merge into exactly the structure we want them, or we basically define by the tissue chamber. And this structure is really this aligned fiber in the center. It's a dog bone structure with two knobs, but the aligned fiber is what we really want. And if we then look into the in a little more detail, we can see that we really have an aligned 3D tissue in this central shaft. And the alignment is not only along on the scale of the previous spheroid, but really it is along the entire axis of the chip. And it's not only structurally connected, but also functionally connected. So if you look, for example, here at calcium imaging, using a GCAM6 reporter cell line, we can see that we really have a fully connected tissue based on these spheroids in the beginning. All right, so these are a couple of ways of how we can generate tissues in an organ-on-a-chip system. The next step, as I mentioned, is the establishment of readouts and also functional validation. I'll show you a couple of examples there. And I'm going to go backwards now. I'm going to start with a hard-on-a-chip. So in the hard-on-a-chip, we added, in terms of readouts and assays, a couple of stimulation and analysis tools. So first of all, we added a simple pacer, and it's actually an open source pacer. We can find Blueprint online. It's very cheap. And the nice part is, in the beginning we thought, okay, let's pattern electrodes in the system for pacing, but we realized we can actually pace our cardiac tissue just by connecting these pacers to the metal connectors that connect our tubings with the chip. And that way we can pace the system, and the tissue will follow nicely up to 2 Hertz. We can then use an optical microscopy-based video imaging using optical flow or block matching algorithm to then analyze the systems without using any invasive readouts. We can do that in parallel. This is, for example, 7 tissues in 1 chip, and we can use that to look at the beating behavior and also some mechanical motions. We can also add oxygen sensors. So we add into the knob an optical oxygen sensor. And this allows us now to look at the oxygen consumption in real time. And here you can, for example, see 2 chambers that are paced at different rates. So you can see you have a baseline oxygen consumption, and the more you pace them, the faster they beat, the more oxygen they will consume. So this gives us now a nice toolbox of different readouts that are all non-invasive that can be used, for example, to test the effect of drugs or to look at cardiac toxicity. The next model is the pancreas-on-chip model. And here we have used first very traditional readouts. So first of all, we looked at low glucose and high glucose conditions and looked at the insulin response. And here you can get the so-called GSIS index, so the glucose-stimulated insulin secretion. And this is very comparable to a well plate. So if you just look at it over globally, you don't gain much to just having these islets in a well plate. However, the advantage is that you can really do time resolved monitoring. So now we sample every 10 minutes or 50 minutes, and you can really see the kinetics of the insulin secretion. So that's one of the examples of the pancreas chip, and even of the initial version. The other example is that you can actually, or advantage is that you can combine it with complex. Here we use Raman spectroscopy, which allows us to do a non-invasive and label-free analysis of the islets. And what we did again, we looked at the low glucose, the Krebs condition, and the high glucose condition, and then took the Raman spectra, and what we could see is that we don't see much difference in the lipid content or the lipid amounts, also not in the DNA or the nuclear amount, but we see a strong difference in the mitochondrial activity. However, when this is in a true component analysis, when we went to principal component analysis, we could also see significant difference in the lipids. So not the amount of lipid changed, but the type of lipids actually changed. So we could see the production of insulin vesicles just using Raman spectroscopy. The other approach we used is, again, to use oxygen monitor. So we have the insulin response of the islet to glucose stimulation, and then we used oxygen sensors in the system to also look at different glucose level at the oxygen consumption. And we could also here see the effect of the stimulation with a higher glucose level. And we did that again by having a small sensor line in the channel, which could be optically, or which can be read out optically. And again, here we can do that globally, but we can also really look at the kinetics of the oxygen consumption. And here you can see whenever you switch to the high glucose condition, the oxygen consumption will increase and the oxygen levels will drop. And as soon as you switch back to the low glucose, it will go back to the baseline. All right, so these are some examples of the readouts and the sensors we can integrate. Now I'm going to show you an example of what we can actually do in terms of dissecting the biology of the system. And I'm going to show you that with the edible system. In terms of the edible system, we also established first a toolbox of different endpoints. And here we looked on the one hand on, let's say, structural assessment. We can show that our adipocytes are really unilocular. They're mature. You see a paralipin A production, but you can also see the functionality. So we monitored the fatty acid uptake by feeding the system with a fluorescent fatty acid. and then using lifestyle imaging, fluorescence imaging to see how these adipocytes take up the fatty acids. And we did that after 1 week, but we can also do that after more than a month. And we can see that the adipocytes are still functional even after 1 month of culture in the system. Then we looked at, for example, the effect of different stimuli. Here we looked at different glucose concentration and the fatty acid uptake. We could look at the fatty acid release if we then, for example, stimulated with isoprotanol, which is a better adrenergic agonist, or also looking at the effluent and looking at the effect of the isoprotanol on the glycerol release in the effluent. So this was a big toolbox which we then use for different types of studies. And one of them really looked into dissecting the cellular interplay, because I showed you that we can have these very different complexity models from the same patient. So from one donor, we can get lots of different models, and we can combine the different cell types in a tailored manner. And what we could see in that case was that, for example, adipokine secretion, which is typically thought to be only produced by the adipocytes. So we see an adipokine secretion if we only have adipocytes in the system. But the more cell types we add, we actually have an increase in adipokine levels. And only in the most complex system, where we have the stromal vascular fraction and the endothelial cells lining the system, we actually have a physiological level of adiponectin or adipokine secretion. The same for other cytokines, for example, IL-8. There you also see a strong impact of the composition of your tissue on the cytokine secretion. So you can really see that this interaction, or the tissue level functionality, is really made up of the interaction or the complex interplay of the different cell types. And it has then secondary effects. For example, if we now perfuse the T cells and use activated T cells, we can actually see difference in the recruitment just by having different types of cells in the system. So that's one of the things we can do with the adipose model. And the other thing is, let's say, our next generation step, where we now combine our adipose models and the liver model. So here we're using a modular slot concept, where you can just use the individual chips and just plug them into this slot. And this enables a flexible connection of different models, in this case, the water and the liver system. And here and this is again, it's a fully thermoplastic system. And what we do here is we can then, for example, look again at the functionality and the structure of the tissue, the adipose tissue on the one hand, but also the liver tissue on the other hand, and line the entire channel connecting the system with endothelial cells. All right, the next model I'm going to talk is our retina on a chip system. In terms of retina, I showed you that we have this physiological complex structure. But how about functionality? And one of the key retinal functionalities is actually a phagocytosis activity, because the photoreceptors are constantly exposed to light, and they have to shed their segments in order to maintain vision. And these segments have to be recycled, and they have to be phagocytosed by the RPE. And so what we could see is that these segments will grow actually into the RPE, and they seem to actually disappear in the RPE after a couple of hours already. And if we looked at it after 7 days, we could find outer segment-like structures inside the RPE in the chip. And after 21 days, you can really see it's one tissue, and you have a strong interaction between the photoreceptors and the RPE, and wherever this interaction is the strongest, you have increased lysosomal activity. So we could really recapitulate this in vivo segment recycling process. And this was crucial for us to show that we really have a functional tissue. And then based on that, we went to some pharmaceutical applications. The first one was rather simple. So we used the systemic delivery of a small molecule. We perfused chloroquine into the system and looked at the retinal toxicity. And we could see a retinal toxicity at high doses, and we could also see the mechanism, the lysosomal accumulation. We then did a long-term study where we looked at lower doses, and we could see that even at the lowest dose, we could see there was already a retinal toxicity. It was a more straightforward application, but we realized that this compartmentalization also allows us to look at other type of delivery. So what we did, we mimicked a subretinal-like treatment, and we added a gene therapy. So we added AAV capsids into the organoid compartment, and now we could look at different types of capsids over long time periods. And we could do that because we separated the nutrient delivery and the drug delivery, similar as you would do it in vivo. And then we looked at the AAV transduction efficacy and the tropism over 3 weeks and could compare different types of AAVs and promoters. And this is now being used by our partners at Boehringer to move some of their candidates forward. So these are two examples of pharmaceutical applications. We have a number of more projects ongoing in that space, but for the sake of time, I won't be able to talk about all of them. What I want to talk about in the last minute is, let's say, the next level of organ on a chip, or what we think is the next level of organ on a chip. Because one of the problems is that the organ on a chip technology promises to combine the advantages of animal and cell culture models. So you want a model that has a high physiological relevance and also a higher throughput. Almost entirely, almost all of the organ on chip research focuses on demonstrating physiological relevance so far. The throughput is rather low. The workflows are very manual and labor intensive. However, this miniaturization, this microfluidic approach actually sets the base for a higher throughput. So we have individual units with sub-millimeter square footprints. So what is needed are scale-up and parallelization and also enabling technologies for automation and user-friendliness. And this is why we came up with what we call the organ disc technology, which combines the concept of organ on a chip with centrifugal microfluidics. And that's how it looks. So we worked with polymer discs, which have cell channels and media channels patterned in them. The cell channels and media channels run radially from the inside to the outside. The cell channels have tissue chambers at the end. And the tissue chambers and the media channels then sandwich a porous membrane, and thereby they are connected fluidically. And what that concept allows us is that we can actually pipette a cell suspension into the central inlets of the disc, and then we take the disc and put it in a CD player, in principle, and when we actually did that, and press play, and thereby all of the cells from the central inlets are being transported into the tissue chambers, and all of that at the same time for all of the tissue chambers around the disc, and it can be more than 100. And now we can use the same concept to actually perfuse this.
Excuse me.
tissues.
Excuse me, Professor Peter. Yeah, we need to bind it up. So kindly, yeah, in 1 minute, please finish.
Yeah, I'm almost done. So in the we can use a media reservoir in the center of the disc and waste reservoirs on the outside of the disc to then actually perfuse the system by rotation. I'm going to jump over that, and because I want to show you how it looks. So this is how the cell loading works. We have actually a number of tissues around the disc, and we can load them at the same time without using any high pressure, and we can actually do sequential loading. And the channels can be lined with endothelial cells and then perfused with media. And we can do that perfusing, as I mentioned, using reservoirs in the center and the outside. We use G forces well below 1G and to get physiological flow rates. And we can also use a different type of perfusion where we add a pump support and pump layer to the center, add steel beads on top of this support layer, then use a magnet. So we don't rotate the disc, but we rotate the magnet underneath the system, and thereby we pull media from the reservoir into the channels and then back into the reservoir, and thereby we have a closed loop circulation. And this can be done again for a wide range of flow rates, and we can use this, for example, to study endothelium, but we can also use this to actually perfuse whole blood through the systems. And we did that in collaboration with Andries van der Meer, and here we studied the adhesion of platelets into the disc or on the endothelium in the disc. With that, I'm at the end. So this is the team that does all that work, and I want to thank also our funding agency, our collaborators, and I want to thank you for your attention.
Yeah, thank you, Professor Loskill. It's a really amazing talk. I think I know audience have many questions, but I request just one-liner questions if anybody kindly come forward.
Yeah. I really like your talk. I had a question about the heart on chip, and I noticed that these hearts on chip, they are missing the ventricle I was wondering if it's relevant and how it can be modeled.
Yeah, so I mean, we use different types of cardiomyocytes. So we have models where we use atrial and ventricular cardiomyocytes, and some of our collaborators actually use this system now to fill it half or to fill, let's say, half of the model with one type of cardiomyocytes and the other side with another type of cardiomyocytes. So you can actually combine different types of cardiac microtissues in there.
Okay, one more question there. Huh?
Hello. Hi, Dr. Subramaniam from Reagent Biosciences. Very interesting talk. Only one comment, question on the organ disc that you have. Is it available, a commercialized product, or still in the development stage?
Not yet. So we are planning actually so we're working on a spin-out based on this organ disc, because we think that's a platform technology where a lot of different organ on a chip approaches could be put and transferred to. So it will hopefully be available as a commercial product soonish.
Okay, thank you.
Hi, this is Indumati here. We work with retinal organoids. Thoroughly enjoyed your talk, and it's great work. So this is regarding the retinal organoids that you were talking about. So you really nicely spelled out the problem of layering the organoids on top of an RPE. By creating a groove, you could overcome the problem. And you said that the maturation does happen very well. So is it uniform across the organoids, and the lamination is it triggered the entire, or just in the region where it is in contact with RPE?
Yeah. So this is absolutely it's a very, very good question, because this is absolutely the limitation of the model right now. The region of interest for us is only the region that is in contact with the RPE. So we see a difference of how the organoid and the maturation of the organoid actually, looks in the region where it is in contact to the RPE when we compare it to the other side, which is nice. We have kind of an intrinsic control, but that's not, let's say, the physiological layering. We are currently working on having the organoids in a flat manner, so that we have the entire, with the layering. And we're making good progress there, but we're not yet there yet. But right now we are really focused only on this region where the photoreceptors are in direct contact to the RPE, and we see a strong impact of that.
Thank you. It's very exciting.
Yeah, thank you. No, the next speaker is waiting, or one more question? Okay, one more question only. Huh. Then, if no more questions, yeah, let us give a big applause to Professor Loskill. Yeah, thank you, Professor. It's really nice. Thank you.
Thank you very much.